Thank you for that introduction. My name is Thomas O'Connor, and to start off our discussion today, it's my pleasure to share some highlights from FDA's research program on characterization of excipients in complex dosage forms. In today's short presentation, I would like to share a few highlights that cover three different areas. First are projects that address challenges related to Q1 sameness assessment for polymeric excipients, for example, PLGA and PEG. Second group of highlights will focus on the development of new in vitro release tests that can be used to determine the impact of excipient properties on complex drug performance. Finally, I'd like to mention some projects that utilize the introduction of new analytical techniques to address polymer excipient characterization challenges. The goal is that highlighting our progress to date that will help frame the subsequent presentations and discussions on the remaining challenges, and what do we need to focus on as we look out over the next 5 years. PLGA is a biodegradable polymer that's used in most long-acting formulations approved by the FDA. PLGA-based injectable microsphere and implants need to be qualitatively and quantitatively the same as the corresponding RLD. However, as a random copolymer, the inherent heterogeneity associated with PLGA makes Q1 assessment challenging. In addition, considering PLGA characteristics can be altered in the manufacturing process, the Q1 sameness needs to be based on the characteristics extracted from the generic product rather than raw material prior processing. GDUFA-funded research have helped address a number of these challenges. Extraction procedures and analytical methods, including NMR-based methods, were successfully established for several key characteristics, including molecular weight, weight distribution, lactic-to-galactic ratio, and end cap analysis. In addition to these characteristics, polymer structure is another critical polymer attribute that can impact product performance. For example, linear or star-shaped PLGA. Research projects help fill this gap, including the development and validation of analytical methods based on gel permeation chromatography with quadruple detection systems that are applied to standards in commercial products. And to achieve the desired release kinetics and application duration, a drug product may contain a mixture of different PLGA polymers. This type of formulation design makes it extremely challenging to characterize the PLGA polymers due to the difficulty to separate one from the other, especially for PGL polymers with similar molecular weight and or different LGA ratios. To overcome this challenge, a research project was conducted to develop methods to separate PLGAs with different lactic acid content based on the solubility difference in different solvents. These are just some of the highlights of the number of advancements in the characterization of PLGA polymers in complex drug products. I also refer you to a review article on FDA's PLGA research program, as well as recent GDUFA research and science reports for additional information and examples. Another polymer excipient that has been examined as part of the GDUFA research is polyethyloxide castor oil derivatives, which are used as a surfactant in some complex injectable drug products. PEG castor oils are available from a number of different manufacturers under varying trade names. They are non-acting surfactants formed by reacting ethylene oxide to castor oil. Depending on the manufacturing process, controls, and ingredient specifications, the chemical structure and corresponding physical chemical properties of these polymeric surfactants can be best described as ranges. How to assess if small variations then in chain lengths, say 5 to 10 monomer units, impact Q1 sameness? The approach adopted focused on characterizing the properties of the PEG. Based on the understanding of the role of the PEG in the formulation, comparative characteristics can be identified. In this case, it was the critical micelle concentration and micelle size distribution in a given formulation. A research project looked at the different PEG grades and found that in the studied range, they had equivalent properties. Other polymer properties, such as molecular weight distribution, etc., may be important depending on the drug product and how it is being used. In both the previous cases, the heterogeneous nature of the polymer posed Q1 sameness challenges. There are also questions and challenges concerning the characterization of the excipient's impact on product performance, both from a quality and bioequivalence perspective. In vitro release tests can be an important tool to compare product performance and discriminate which material properties and process parameters impact performance. IVRT can be especially important when how the excipients interact with the manufacturing process impacts performance. The FDA research program has advanced a number of novel IVRT methods to support the characterization of complex products. In one example, highlighted here, adaptive fusion, a pressure-driven separation method based on the principles of tangential flow filtration, was developed for investigating the rate and extent of drug release from drug products containing particulates, such as emulsions, suspensions, liposomes, and drug protein complexes. The adaptive perfusion method achieves size-based separation of the particulates, while simultaneous analysis of the released drug, as well as the remaining drug, and the method can be optimized based on the formulation by adjustment of the filter, molecular weight cutoff, feed flow rate, or back pressure. Notably, the adaptive perfusion method provided discriminatory drug release profiles for drugs in solution, micelles, and in small, medium, and large globule size emulsions, and in a significantly faster time frame. The IVRT method has the potential to be extended further to examine the impact of the manufacturing process on drug distribution and release characteristics of other challenging complex drug products. For example, formulation components can be selectively retained based on molecular weight cutoff range of the membrane. In this case, castor oil globules, nanoemulsions, or protein-bound drug containing nanoparticles. The selective retention process can effectively help in differentiating the rapid drug release from the extended phase drug release. Complex excipients, such as polymers composed of hydrophilic and hydrophobic monomers, can be selectively retained based on molecular weight by using the adaptive perfusion method. The impact of these polymers on drug release can then be studied, including compositional or manufacturing process changes, and evaluating their effect on drug release of the resulting product. In another example from the GDUFA-funded research program, a non-sync discriminatory in vitro release testing method was developed, which combined in situ fiber optic UV with laser diffraction to simultaneously monitor the dissolution and changing particle size distribution of a suspension. Injectable suspensions occasionally exhibit variations in dissolution bioavailability, which may impact the clinical outcome of the drug product. Drug release in these injectable suspensions is controlled by the formulation design, including API particle size. Notably, suspensions are often designed to weakly flocculate, which increases the long-term product physical stability. However, as flocculation inherently influences the effective particle size of a suspension, the impact on dissolution and consequently bioavailability and therapeutic effectiveness of the product should be considered. Particle flocculation is a complex phenomenon that arises from interplay between interfacial chemistry and environmental conditions that govern particle-particle interactions. In the suspension formulation, this process of agglomeration is controlled by the use of wetting agents, typically surfactants, and polymeric excipients for steric or electrostatic repulsion, and by varying the environmental conditions, pH and ionic strength. This is a reversible process where the breakup and redispersion of particles is impacted by the shear stress imparted on the system. In this study, the variation in the injection method, I applied shear, for triclonal acetate and injectable suspension, altered the flocculation state of the particles and subsequently their dissolution. The IVRT method developed simultaneously measured the dissolution and the particle size distribution and showed that flocculated and deflocculated particles follow different dissolution pathways. Importantly, deflocculated particles dissolve up to 6 times faster than flocculated particles. Similar shear-induced changes during injection could occur in the clinical setting, have implications for drug bioavailability. The final area that I would like to highlight is the introduction of novel analytical methods to address characterization challenges. AF4 belongs to a class of field flow fractionation techniques which are diverse among each other, each of them being more suitable for a specific purpose. But they are all based on the same principle. Solutes fractionate among layers of a continuous flow under the influence of an external field without the presence of a stationary phase. The main advantage of these techniques are a wider operational range as a number of experimental factors can be tuned, superior sizing accuracy, compatibility with numerous types of detectors, and greater separation power, especially for polydiverse dispersed samples. In this project, AF4 was applied to characterize high molecular weight polyethylene oxide, which has been used in solid oral opioid formulations for abuse deterrent properties. High molecular weight PO is not stable and may degrade under conditions that may be experienced in the manufacturing and manipulation process. The molecular mass is an important property affecting the properties of the PO polymer, where the high molecular weight and polydispersity present characterization challenges. Traditional characterization methods such as viscosity measurement and size exclusion chromatography have limited resolution and dynamic range, so a new characterization method based on AF4 was developed. The AF4 method developed considered both the conditions needed for low and high molecular weight PEO and implemented a gradient cross-flow program to achieve this. It was found that the AF4 can serve as an orthogonal method for size exclusion chromatography for PEO that are less than 1 million megadalton. And if AF4 provided better characterization results for larger polymers compared to SEC. But there were still some challenges noted during the study for ultra-high molecular weight region, which may require further method development to address. We have also applied AF4 to other challenging problems, including the characterization of emulsions globule size distributions. While I was only able to mention some highlights over the past few years from the research program, I think there are some general themes to addressing scientific challenges posed by the characterization of excipients in complex drug products. First is understanding the properties of the excipients and their role in the formulation can inform the characterization approaches. Then, depending on the complexity of the material attribute that needs to be measured, novel analytical approaches may be needed. Then, in addition to raw material properties, manufacturing and post-processing can impact the microstructure and performance of the complex drug product. This leads to the need for discriminatory in vitro release tests that can facilitate the assessment of the impact of the manufacturing process and post-processing on the microstructure. In closing, I would like to thank everyone, both in the FDA and across the network of external collaborators who have contributed to the advancement we have achieved to date. And I look forward to discussion on the remaining challenges and where we should focus our efforts going forward. Thank you.